form of cancer, which is the invasiveness or metastasis. They function as a DNA repair mechanism. There are what is called caretaker and gatekeeper genes. So here they are caretaker genes encode protein that repair the genome mutations. As we mentioned, cell cycle is not a perfect biological function. During it's very complex, and during this complex process of replicating the cells, replicating the genome, the cell organelles, all this leads to a chance of damage or errors to the organelle or DNA damage. So this is tumor suppressor genes can help as a caretaker genes, prevent replication of cells with mutations. So they can sense if there is damage to the DNA and therefore preventing cell replication and eventually apoptosis. So it has its own mechanism of repairing the genome. Now we'll take two examples of the tumor suppressor genes. The first one is known as retinoblastoma, or RB gene named RB capital, protein named RB small. So they prevent cell or excessive cell growth. They are also a gatekeeper gene that block cell proliferation. We mentioned gatekeeper and caretaker. Caretaker take care of the damage, but the gatekeeper block the proliferation. There it allows try to repair the damage for the caretaker, but here in the gatekeeper it blocks the cell proliferation. Prevents cell cycle specifically. Prevents cell cycle progression from G1 into S phase. So this is the first transition, making sure that the cell has the sufficient nutrients, sufficient growth factors, sufficient material to start synthesizing the DNA in the S phase. So here it works. Here it prevents the cell cycle from progressing until, of course, the conditions are met. DNA damage, if there is a DNA damage, then retinoblastoma bind to a transcription factors known as elongation 2 factor. This will repress necessary gene transcription, preventing cell from replicating its DNA, as we'll see it in the next diagram. So here again, the retinoblastoma tumor suppressor gene are negative regulator, are brakes. They prevent, they repress. This is how their function is opposite than the CDA cycle-independent kinases. And of course, they work together in harmony. Now, here's what happened, how the retinoblastoma protein activity is regulated. Here we see the gene needed for S phase is not transcribed. Now, let's say the cells are not in the phase that's going to transit into S phase. So we see that retinoblastoma bound to the elongation factor. In this case, there is no transcription. So this is the inhibited form of elongation factor. Transcription factor is not working because it's inhibited by retinoblastoma. Now, when the signal comes, growth factor, RAS pathway, whatever the signaling pathway in the cell cycle-independent kinases, it will tell the cell now we need gene transcription because now the cells is going through S phase. So we need synthesis. We need to remove the brakes. So what happened here, that the cyclin-dependent kinases, it phosphorylate retinoblastoma. See, retinoblastoma here now is phosphorylated at 2 sites. Who phosphorylated them? The cyclin-dependent kinases. Which one? The specific combination that require the transition into S phase. Right, so now once it's phosphorylated, it's unbound. When it's unbound, because it's a negative regulator, now elongation 2 factor allowed to transcribe genes that are required for the transition into S phase. All right. So this is, again, you see the complexity, they work together at different layers. Cyclin-dependent kinase doing things, and then it works on inhibitory doing things. Together they work in coordination to allow the activation and deactivation, of course, by phosphorylation. When cell is ready, when the cell is ready to divide, retinoblastoma is phosphorylated into phosphorylated RP. This is when ready. What we mean by ready, all the conditions are met: cell size, cell nutrient, growth factors, genetic materials, cell size, and so on. All, and there is no damage. There is no damage to the genetic materials. Then RP is phosphorylated to the phosphorylated form, and therefore the transcription can proceed, leading to inactivation of RB activity and therefore gene transcription. Because they are breaks, we need to inactivate them in order to allow the transcription to happen, or desuppression, desuppression in order to allow activation of the gene transcription for the cell cycle to proceed. So this is how they work together, two different components, two different layers doing different things, but all are linked to the condition of the cell and the presence or absence of damage. So what happens if we lose the function of retinoblastoma? Multiple responses in different cells ultimately could result in cancer. Specifically, there are certain things that the loss of function mutation in RB could result in. Decreases the differentiation potential of cycling cells. It means that there are certain cells that are differentiated at some time, but this differentiation, these cells usually will be terminally differentiated, unlikely to re-enter the cycle. However, if the retinoblastoma is mutated, there is a decreased differentiation, means that the cell will continue in the cycle, will continue to duplicate, multiplicate, proliferate. There is decrease in differentiation and increase on the other side of cell cycle re-entry in differentiated cells. So if some of the cells were differentiated, mutation or retinoblastoma induce them to enter the cell cycle or cell cycle re-entry of differentiated cell, and they will continue to replicate as undifferentiated cells. Prevent induction of cellular senescence. Senescence is what? Cells become old, become functionally not very active or optimum. So usually the cell has mechanism to induce senescence. Senescence become quiet, become less function, but preventing the error or the problems from entering the mainstream replication or division. So here it prevents induction of cellular senescence, means that the cells are actively dividing, which means with a lot of activity inside the cell when they're dividing, there are a lot of potential of errors and mistakes and damages to the genetic material. Increased chromosomal instability. This is why, because of the damage. There are DNA damages that cannot be repaired because retinoblastoma now is mutated, loss of function. So the DNA damage check is not appropriate. Increased chromosomal errors, damages, and therefore instability. Increase metastasis potential زي ما ذكرنا علاقتها بالloss of contact adhesion, and of course promote angiogenesis in the tumors. So all these specific functions has to do with the life cycle of the cell, mechanism in the cell cycle where the loss of function of retinoblastoma can induce, and all of these collectively can lead to tumorigenesis. Now we'll get to the other one. Similarly, somehow, tumor suppressor genes called tumor protein P53. It's also implicated in a lot of number of cancers and tumors. It's known as the guardian of the genome because its mechanism is really complex and it has multiple functions along the side of retinoblastoma. It regulates cell cycle, apoptosis, and genomic stability. Activate DNA repair protein when DNA is damaged. This will arrest the cell cycle at G1S to allow for DNA repair. And we don't want to go to the S phase unless we have genetic materials, we have all the DNA materials are the right way, the right amount and there is no damage. So if the DNA is repaired, then the cell cycle will continue. If irreparable, initiate apoptosis. So this is somehow similar to retinoblastoma. Now, a lot of similarity between TP53 and retinoblastoma or RP genes. Now, in terms of the function, also there are certain mechanisms that allow the activation or deactivation of P53. Usually, P53 is bound to a protein